Hello and welcome to my channel. Today we'll be discussing the histology of the nervous system, so let's start. Cells of the nervous system, of course, basically it's the neuron, which is composed of a cell membrane, which is a bilipid layer with hydrophilic ends towards the outside and the inside. Means towards the outside and the cytoplasm, and the hydrophobic ends of the bimolecular layer is in the middle. Contains also proteins that make up the channels in the membrane. The cell body, called the perikaryon or soma, it has neurites, which are processes extending from the cell body, either dendrites or axon. And it's the purpose of which to increase the surface area. The total volume of the cytoplasm in neurites is more than the cell body cytoplasm. So if you calculate the amount of cytoplasm in these neurites, it's more than the amount you see in the cell body. The nucleus has a double layer membrane with pores, has DNA, chromatin, nucleolus, which is made up of RNA for RNA synthesis. The bar body, it's one of the two X chromosomes by inner surface of the nuclear membrane. The cytoplasm contains Nissl bodies or Nissl granules. They are RNA granules, ribosomes. They are present all over the cell body except in the axon hillock, which is the start or the beginning of the axon. Remember that the axon hillock is devoid of the Nissl granules. The cytoplasm also has mitochondria, has a Golgi apparatus, which are vesicles of a smooth endoplasmic reticulum for processing and packaging around and present around the nucleus. Also has lysosomes that formed from Golgi. They are membrane-bound vesicles, contain hydrolytic enzymes to act as scavengers. The cytoskeleton is made up of mainly of microtubules, neurofilaments, and microfilaments. The cytoskeleton responsible for the shape and transport in neurons, and this is important, transport in the neurons. It forms like railroads, for structures to protein carriers move structures on top of it. Composed of microtubules and microfilaments and neurofilaments, as I said. The microtubules you see here is a microtubule composed of 13 linear protofilaments, and each made of pairs of alpha and beta subunits of protein called tubulin. The neurofilaments, they are coils of thin protofibrils present more in the axon and change into what's so-called the neurofibrillary tangles in Alzheimer's disease. They are used to diagnose Alzheimer's if the neurofibrillary tangles are present. Microfilaments, this is a microfilament as you can see here, are strands of polymerized globular actin monomers arranged at a helix, important in motility during development, and maintains the shape of the cell. So remember, microtubules, neurofilaments, and microfilaments. Now the dendrites are short. They taper distally, as you can see here. They are present to increase the surface area for reception. Dendritic spines, to further increase the surface area, there are dendritic spines from branches to increase the surface area. Cytoplasm is similar to that of the cell body, but without the Golgi. The Golgi present around the nucleus. And the dendrites only transmit signals. They do not generate the action potentials. The action potential is generated in the axon hillock at the beginning of the axon. Dendrites, they do not generate action potentials. Now the axon, you see here the axon, single, cylindrical and long. It has a cytoplasm called the axoplasm and the membrane called the axolemma. It's covered with myelin, but the myelin is interrupted at nodes of Ranvier, and present at these nodes of Ranvier are the condensation of sodium channels, but the potassium channels mainly present underneath the myelin. So that's why the impulse travels from node to node. Okay? Contains mitochondria, the axon, microtubules, and neurofilaments. The axon hillock, as mentioned before, is the site of generation of action potential, and it's devoid of Nissl granules. The axon doesn't branch except giving a collateral when it leaves the cell, but branches only at the end, called terminal branches, and each terminal branch has a synaptic terminal, which is called the synaptic buton. See the terminal branches of the axon. Now in the axon, transport of substances occur in the axon in both directions, away from the cell or away from the soma and towards the soma. Away from the soma is called the anterograde transport, and towards the soma is called the retrograde transport. Now, in each transport, there is a fast and slow types. The fast anterograde transport is forward flow. It's for the transport of needed substances like the precursors of the neurotransmitters or enzymes or building blocks to repair structures, sites in the cell, and glycoproteins for this is reconstitution of plasma membrane. Rough endoplasmic reticulum forms the structure to be transported, packaged by the Golgi apparatus into vesicles, and the vesicles attached to microtubules. I told you before, they act as railways. Movement occurs by alteration in an enzyme called, protein enzyme called kinesin ATPase. Gets its power from the hydrolysis of ATP. Then the vesicle formed is cleaved and exits the cell by exocytosis. Now, surplus elements from the vesicle, when it empties its contents, go back in retrograde transport for recycling or degradation by lysosomes. Slow anterograde transport occurs in the movement of microtubules and neurofilaments, actin, myosin, calmodulin, which is a calcium binding protein, and tubulin. It's the speed of which is much slower than the fast anterograde transport. Now, there is fast retrograde transport, which although it's fast, but it's slower than the fast anterograde transport, and it's for transport of surplus membranes, nerve growth factors. This is important in the development of the nervous system. It depends on nerve growth factors, so picked up by the cell and transported by the fast retrograde transport. It stimulates the growth of neurons, and substances taken up by endocytosis travel also by fast retrograde transport. How can we apply this in pathology? There are retrograde viruses like the herpes simplex virus, the polio virus, and tetanus, and that's why they take some time to develop symptoms, because they infiltrate the neuron at its periphery and then transported by retrograde transport. Also, as an application, we can use this phenomenon as tracing techniques. With microinjection of fluorescent dye like fluorogold, we can trace different. This is the way, by the way, they trace tracts in the spinal cord and tracts in the cerebrum. Now, types of neurons. And remember, there's a difference between neurons and neuroglia. Neuroglia are support cells. Neurons are the principal units of the nervous system. There is multipolar neurons, as you can see here. Multipolar have many dendrites and one axon. Present as giant multipolar neurons in the anterior horn cells of the spinal cord, and it takes the shape of a pyramidal in shape in the cerebral cortex, especially the motor cortex, the Betz cells in the cortex. Purkinje cells, these are present in the cerebellum. They resemble the giant, the pyramidal cells in the cortex, and they are flask-shaped, many dendrites in the cerebellum. Also stellate cells in the granular layer of the cerebellar cortex, and we will study this when we study the cerebellum. So, multipolar neurons, examples are pyramidal cells, Purkinje cells, and stellate cells. Remember this. Bipolar neurons, this is a bipolar neuron, means that from one end there's an axon and the other end there's a dendrite, and it's present in olfactory mucosa, in the retina, and the spiral ganglia of the inner ear. When if you remember, when we studied the inner ear, we said that the cochlear nerve takes impulses from the organ of Corti and then go to the spiral ganglia in the modulus of the cochlea. They are made up of bipolar neurons. Pseudo-unipolar neurons. Pseudo-unipolar means like a unipolar, but it's not. It's globular in shape, as you can see here. Have one process that branches into an axon, and so this is the process that branches into an axon and a dendrite. And the pseudo-unipolar neurons present in the dorsal root ganglia, in sensory ganglia of cranial nerves, especially the mesencephalic nucleus of the trigeminal nerve. So remember this, the pseudo-unipolar neurons present in the dorsal root ganglia. They are the neurons that make up the dorsal root ganglia and the sensory ganglia of cranial nerves. Unipolar neurons, these are traces in human beings, but present in invertebrates, and characterized by the fast response. Marine life, the creatures change direction in very fast speeds depending on unipolar neurons. The neuron have one process called a neurite and extends from the soma, then branches to form dendritic and axonal processes, the unipolar neurons. These are the types of neurons present in human beings. The gray matter is made up of the soma of cells and the unmyelinated axons of neurons, and it's present at the outside layer in the cortex and inside mass in the spinal cord. Now, the white matter are the axons and dendrites. It's mainly axons is myelinated fibers inside the brain and present outside mass in the spinal cord. They form communication between neurons through synapses, which are either chemical or electrical. This is a chemical synapse, as you can see here, and this is an electrical synapse. Now, the chemical synapses, they have a presynaptic membrane. See that? Cleft between in the synapse, and then postsynaptic membrane that contains receptors for the neurotransmitter released by the presynaptic membrane. The presynaptic membrane has mitochondria, provides energy for the neurotransmitter release, vesicles of stored neurotransmitters, and the release of these neurotransmitters from their vesicles depend on calcium. They are responsible for the rupture of neurotransmitter vesicles. And common forms of synapses are axodendritic, axo-axonic, and axosomatic. Less common forms are dendritodendritic and dendrito-axonic. Electrical synapses, the principal structure is a connexon, structure called a connexon, and they form gap junctions between the cells for ion passaging. See, ions pass through these connexons, and it's faster than the chemical synapses. So the way neurons communicate by synapses, either chemical or electrical synapses. Now, for the connective tissue cells, which is called the neuroglia in the nervous system, they are support cells, and they are not excitable. Means they are not they don't function in propagation of the nerve impulse, and more numerous than neurons, 5 to 10 times more. They are composed of astrocytes, oligodendrocytes, microglia, ependymal cells, Schwann cells, and satellite cells. If you take a look at the astrocytes, this is an astrocyte. Its star-shaped processes extend in all directions, as you can see here. The functions of astrocytes, they are support neurons, provide nutrition, means it provides nutrition, and forms barrier against spread of the released neurotransmitters, so as the impulse not to spread all over the nervous tissue. Forms an insulation that prevents electrical activity to affect nearby neurons. Also picks up some neurotransmitters like glutamate and gamma aminobutyric acid. Picks up potassium ions after local neuronal activity, and during development they secrete trophic factors for growth of neurons. They also have what's so-called podocytes at the end of their processes that forms the blood-brain barrier that join to form a glial-limiting membrane or perivascular lining membrane, and they are responsible for scar formation and filling the gaps in injury of the nervous system. Types of the astrocytes, according to their location, the ones present in the gray matter, they are called protoplasmic. They have thicker and shorter processes with many branches. The ones present in the white matter, they are called the fibrous astrocytes, and they have thinner and longer processes with less branches involved in repair and damage of the nervous tissue to form a scar formation. In the retina, there are modified types of astrocytes called the Muller cells. This is the astrocytes. Oligodendrocytes, they are myelin-forming cells. They are smaller in size and have fewer and shorter branches. They don't have neurofilaments and myelinates multiple axons in the CNS. Remember, in the CNS, the cell that myelinates the axons in the CNS, which means inside the bones, inside the skull or the vertebral column, means the brain and spinal cord, are oligodendrocytes. The problem is that the oligodendrocyte myelinates more than one axon, so a damage to oligodendrocyte involves damage to many axons that it myelinates. And they are affected in demyelination diseases. Microglia, they are small and have short thorn-like processes. The only cell that derive from mesoderm, like a blood, like cells of the blood. It's an antigen-presenting cell and has phagocytic function and is activated in injury. It enlarges and becomes mobile and phagocytic. Schwann cells, they myelinate the peripheral nervous system. Okay, so the myelinating cell of the central nervous system is oligodendrocyte, while the myelinating cell of the peripheral nervous system is Schwann cell. Okay? Satellite cells, the cells you see here, they are called satellite because they surround the ganglia, especially they surround the pseudounipolar cells in the dorsal root ganglia. It's very obvious here. Other type of cells, they are called ependymal cells. They are related to the CSF circulation. They are composed of three types: ependymal cells, tanycytes, and choroid epithelial cells. The ependymal cells, the cells you see here, they are cuboidal and columnar cells. They line the ventricles of the brain and spinal cord, central canal, and they have microvilli of absorptive function and cilia for the flow of the CSF. As we said before, ependymal cells, they are related to the CSF circulation. Tanycytes, they are ependymal cells, but modified ependymal cells present in the third ventricle. It has processes that extends into the brain tissue to blood vessels and neurons, and it's responsible for transport hormones from the CSF to capillaries of the portal system. Other type of ependymal cells is the choroid epithelial cells. They are modified ependymal cells and present in choroid plexus that's responsible for production of CSF, and have tight junctions because it forms what's so-called the blood-CSF barrier. So remember, there is a blood-CSF barrier and there is a blood-brain barrier, okay? Ganglia of the nervous system, they are subdivided into sensory ganglia, which are pseudounipolar neurons and present in the dorsal root ganglia and sensory ganglia of cranial nerves. Autonomic ganglia, they are made up of multipolar neurons, so please don't get confused. The pseudounipolar cells form sensory ganglia. The autonomic ganglia is made up of multipolar neurons present in sympathetic ganglia and parasympathetic ganglia, like the Ehrbach's plexus and myenteric plexuses in the gastrointestinal tract. And as a general plan, the nervous system is anatomically classified into central nervous system, inside the bone, means inside the skull, the vertebral column, which is the brain and the spinal cord, and a peripheral nervous system, which are the nerves that coming out of the central nervous system. And these nerves, whether they are somatic or autonomic, they are classified as peripheral nervous system. And functionally, if you the functional classification is into somatic, voluntary, and autonomic, which is involuntary systems. I hope you got the maximum benefit from this lecture, and thank you very much for watching.